Well, hi again, and again, my apologies for having to deliver a lecture, another lecture by screen recording, but thank you for your patience as you bear with me. We'll be talking today about microbial diseases of the skin and wounds and hopefully get into some similar conditions of the nervous system near the end of the lecture today. So for the last several, lectures now, towards the end of the course, this is what we focus on, our specific pathogens and corresponding diseases. We'll spend time on those particular types of diseases which may exemplify certain important principles or are of otherwise interest historically or looking forward in the prospect of emerging diseases. So as we begin the topic of skin and wounds, you want to make sure that you review the basic principles of the anatomy of the skin. Remember, this constitutes an important first line of defense. The innate immune system, and this diagram in your text shows you is an illustration of the structure of the epidermis and dermal layers of the skin, reminding yourselves that the microbiota in the upper layers of the epidermis are part of the innate immune system as they help prevent colonization by potential pathogens. And hopefully now that you recognize the role of these guys here, these dendritic cells, again, these will sample organisms associated with the skin. These can then travel to lymph nodes where they can present microbial parts then to T-cell lymphocytes. So that's part of the an adaptive immune response. So make sure you understand just the basic structure, the epidermis, the dermal layers which underlie that which is vascularized. It's too bad we didn't have lab last night. It's interesting to see the numbers of microbes that still do remain after vigorous hand washing. And that really drives home the point that microbiota are associated with the hair follicles and other layers which they're just simply not as easily removed as the transient microbiota are. So the cells also have tight junctions, which means they're attached very closely to prevent introduction of microbes, although, of course, wounds, which can occur by a number of means, can allow the microbes to infect deeper tissues of the body. So if you think about why the surface of the skin is not a good habitat for most microbes, review these principles, that there's these antimicrobial substances secreted by both the epiderm, both by the sebum and the dermal cells, as well as by members of the normal flora. Dermacidin is simply a general name typically for small peptides with antimicrobial activity. Fatty acids can also exhibit antimicrobial activity, some of these serving to lower the pH. So the low pH is not tolerated by many microbes. The saltiness of the skin is another inhibiting factor, as well as we've mentioned a number of times the normal flora. So this, I believe, comes from another textbook, but this kind of summarizes the features of the skin in various parts of the body and their role in protecting invasion by microorganisms. This is a figure that we covered back in another lecture on the microbiota of the human body, but seeing here we're focusing today on the skin, mainly composed of the normal flora, the actinobacteria. So those include the dipteroids that your text refers to. Dipteroid is kind of not really a taxonomic term, but more of a general term to refer to members of the actinobacteria that have sort of strange or irregular cellular morphology. And those include Corynebacterium and Propionibacterium. So both of these genera can also occasionally be involved as opportunistic pathogens. But again, if you hear the word dipteroids, that's what those refer to. A little bit more minor component, the Firmicutes, that includes the Staphylococcus. So these are gram-positive bacteria. and Staphylococcus and Micrococcus are two common members then of that group that would be found on the skin. Oops, let's go back here. So again, the lab last night would have demonstrated that most of these members can't be completely removed even with thorough cleansing, but we'll decide whether or not that's really an issue in a healthcare setting. So occasionally, even though the primary function of the normal flora is to protect us, they can occasionally be involved in diseases if they're able to gain access to parts of the body where they would normally be excluded from, or if the immune system is depressed. Simplest type of skin infection, which is folliculitis, simply infection of the inflammation of the hair follicle, causes pimples on the skin or styes if it occurs on the eyelids. Now normally these resolve quickly, as you're probably aware. White blood cells, as in this case, are able to infiltrate the infected follicle to clear the infection, although in some cases the infection can spread to form boils or even larger raised lesions called carbuncles. The initial infection occurs when the follicles become clogged with dead skin cells and sebum, allowing the accumulation of fluids in the follicle and a good environment for growth of bacteria such as Propionibacterium, which causes acne, and we'll get to that a little bit later in the lecture, but more commonly Staphylococci. So Staphylococci are by far the most important genus of bacteria involved in skin infections. So we're going to spend a little bit of time talking about this genus in some depth. Now, as we go through these diseases, I want you to focus on what are the bacteria, what are the major species involved in the different conditions, and what are some unique characteristics of each of those diseases. You do not need to focus, unless I indicate otherwise, what the various treatment options are, because these, of course, are always changing. There's evolving standards of care, and of course, with the evolution of resistant strains, things like treatment can change quite readily. But do understand what the major causes are and what some of the features of the development of the different diseases are. So Staphylococci, something that you will encounter, I'm sure, quite often in your career. These are also, these should be familiar to you from some work we've done in lab already. Know that they are gram-positive cocci that typically form these grape-like clusters. So this is a diagnostic arrangement. This particular sample came from pus from a skin lesion. They are, of course, tolerant of salt and the dry conditions found on the surface of skin, so they may be commensals that then become opportunists, or in other cases, as in Staph aureus, they may be transient and then can initiate the disease by virtue of various virulence factors. So of particular concern are MRSA strains or methicillin-resistant Staph aureus. Most of these can still be treated with vancomycin, but of course prevention should be a priority. So hand washing and other types of antisepsis to prevent transfer of staph between different patients. There are actually two species, though, of staph which are of particular importance in a clinical setting, and the first of these Staph epidermidis you've worked with in lab. This is a BSL-1 pathogen, which means that it almost never causes disease, although in a clinical setting they can colonize various medical indwelling devices such as urinary and venous catheters. So because they exist as a significant component of the skin microflora, during insertion of these, they can be picked up and then establish themselves as biofilms. But by far, Staph aureus is of much greater concern, both because of the variety and severity of the diseases that it causes. Now, it's not a significant or even universal member of the normal flora, although about 20% of people do carry pathogenic staph as a commensal. This is not surprisingly a concern because it can be transmitted to others, including in a patient setting by direct contact or fomites. Fomites, as you recall, are inanimate objects that one handles and then somebody else picks up and acquires the pathogen indirectly. Now, under a microscope, these two species look the same, but they can be differentiated pretty easily in the laboratory by their differential appearance on agar. So you'll recognize staph epidermidis is forming these little white colonies. In the lab, here on blood agar, there's no evidence of lysing of the blood cells, but clearly we haven't used this in the lab, but Staph aureus, if it was grown, when it is grown on blood agar, completely lyses the red blood cells in the area surrounding the colonies, and this again is called beta hemolysis. On media such as mannitol salt agar, the differential ability of Staph aureus to ferment the mannitol produces acids and then a yellowing of the media, so that allows differential allows them to be distinguished from Staph epidermidis by that characteristic. Again, the salt in this particular medium prohibits the growth of non-staph species. So let's review again how the types of, in this case, virulence factors, which allow Staph aureus to be invasive. It very often causes infections involving production of pus in superficial skin infections. And the pus is really the accumulation of the contents of lysed host cells, cytotoxins produced by Staph aureus that lyse red blood cells, hemolysins, and white blood cells, leukocytins, are involved, as well as other enzymes such as DNase. So that contributes to the pus formation, and then enzymes such as coagulase, which we've mentioned before, forms clots then that wall off the initial site of infection from cells of the immune system, such as phagocytes. Coagulase negative staph are much less virulent because they're unable to form that protective barrier. So that's illustrated in this figure on your slide. One thing we have not talked about yet is the concept of superantigens. So here's my little rabbit. We're going to go off on a rabbit trail and talk about superantigens because they're such important virulence factors of not only staph but of strep. And it didn't really make sense to talk about these until we had a pretty basic understanding of how the adaptive immune system works. By the way, this is an actual isolate of staph of MRSA, methicillin-resistant staph, showing that for now this is still sensitive to vancomycin and thus could be treated with this antibiotic. Okay, so superantigens. You'll recall from the chapter on adaptive immunity that when phagocytes at the periphery, which means at the surface of the skin perhaps, the dendritic cell, specialist type of phagocyte, ingest microbes, that those are processed and then presented on MHC molecules, which is the major histocompatibility complex. So the antigen in this case, which is the part or subunit of the microbe, which the phagocyte is alerting the rest of the immune system to the presence of the infection, it's again on this MHC molecule. It migrates to the lymph node, and at that point a T cell, which has a complementary T cell receptor, forms a synapse. So it requires that the antigen in this case be complementary to both the T cell receptor as well as being presented on the MHC molecule. So this is how antigens are normally presented in a much even more simplified diagram here. The T lymphocyte recognizes this particular antigen and it requires the presence of the antigen here. Superantigens are bacterial toxins that form non-specific synapses. Essentially, they take any antigen-presenting cell with an MHC class II molecule and non-specifically attach it or activate a T cell. So the Superman symbol there is indicating that this is occurring even without the presence of an antigen. So this is an abnormal situation. In a little bit more detail here, on the left side is how, again, the process normally occurs where, in this case, a viral epitope here is presented on an MHC cell to cause specific activation of the corresponding T cell. The result then is that the cytokines which are released cause this specific T cell to multiply, and then the receptors, the T cell receptors here, have a very specific affinity to the antigen that elicited that response. So the result is a very strong antiviral activity, specific immune reaction. Well, what about the case of a superantigen? So say the superantigen, and again, these are types of toxins produced by staph, strep, and also some viruses, as in this illustration. This superantigen may produce, the virus may produce this molecule, the superantigen showing here, which connects this antigen-presenting cell with a T cell, but it's a non-specific T cell. And it does this to lots of different T cells, and the cytokines which are released cause then a polyclonal as opposed to a monoclonal activation. Only a small subset of these will then have any affinity or specificity to the infectious agent. So a very weak and non-specific immune response occurs.